So first we'll look at random sampling. As its name suggests, random sampling is really just the random selection of quadrants. It's a very, very common method in ecological studies. It's used to assess the abundance of species in a given area using some of those metrics we saw earlier. So think species richness, species evenness, percentage cover, etc. This technique basically involves dividing the area of interest into equally sized quadrants, so those quadrants we saw earlier, and then using things like a random number generator or some means of randomly allocating these quadrants to select a random percentage of quadrants to be used for sampling. By focusing on randomly selected sections, researchers can gather data that is representative of the larger area while minimizing potential biases. And obviously this does save us a lot of time for actually just getting an entire ecosystem, which is somewhat unrealistic. One of the primary advantages of random sampling is it allows us to use a couple of statistical tests, many of which assume that the data is collected from a random sample. This just basically increases the reliability of our results or our view of the ecosystem, and in particular enables researchers to draw more accurate conclusions about the abundance of species in an area. Additionally, random sampling obviously minimizes bias and human error, as the selection of quadrants is not influenced by the researcher's preferences or preconceptions. If you were to look at an ecosystem and there was something really interesting happening in the top left corner, your interest might be to plot a quadrant there. But in doing so, you're not giving an accurate representation of the entire community. But random sampling does also have its disadvantages. One significant limitation being that it doesn't account for microhabitats or areas with different biotic factors. Obviously, like we saw when we're looking at the spatial scale, ecosystems can vary incredibly, even within themselves. So doing this is obviously ignoring a whole heap of the ecosystem. This means that important variations within the ecosystem, such as small patches with unique environmental conditions, like I was discussing earlier, even though they are randomly overlooked, they are still overlooked, which can lead to a complete misunderstanding of the ecosystem's actual biodiversity. Another drawback is that random sampling doesn't consider temporal changes, which can be crucial in understanding ecosystem dynamics. For example, nocturnal animals or seasonal variations in species presence and activity are obviously not going to be captured by a single sampling session. You'll have to do multiple measurements over time in order to capture them. So those are the main disadvantages we see. So we also have another countermeasure that might try to address some of these, which is systematic sampling. Basically, this method is used in ecological studies to assess specifically species distribution and abundance. It involves dividing the area of interest into quadrants and then systematically allocating which quadrants to study. So for instance, by sampling every third or every fifth quadrant, we're able to get a more even distribution of the ecosystem. It provides a bit more of a more structured and organized way to collect data on a large area, and again, it ensures that the sample is evenly distributed, which is the main advantage it presents. Another main advantage it has is its efficiency, quite like random sampling. By following a predetermined pattern, however, researchers can cover a lot of area in a set amount of time, and they don't have to go about trying to randomly select what area to do and making sure that they are covering the right areas. So that makes this method particularly useful in extensive or really difficult to access ecosystems. Additionally, systematic sampling minimizes bias and human error because the selection of quadrants is based on a fixed system rather than your subjective choice. So just like with how we saw with random sampling, you can't implement bias in order to determine what quadrants you'll look at. And this basically just helps us to ensure that the data collected is consistent and that every part of the study has an equal chance of being represented. But much like our random sampling, this method also has some limitations. One significant drawback being that statistical tests really do not account for this type of sampling. They usually expect random sampling. That's just because of how they work. They expect a random sample in order to graph accurately. So the structured nature of systematic sampling can limit the types of analysis that can be conducted on the ecosystem. So it can be a little more difficult to classify an ecosystem as some of the ones we saw earlier, such as old mature forests, coral reefs, and so forth, since we have less statistical tests to prove what they are. Another limitation is that systematic sampling doesn't account for temporal changes or subregions within the area of interest, just like random sampling. So that means, again, variations in species presence or activity that occur at different times of the day or in specific microhabitats can be easily overlooked with this method. So again, that leads us on to our next technique, which is probably the most common one you'll see. We'll look at this method in the context of some key techniques that you'll be using: line transects, quadrant sampling, and belt line transects. So stratified sampling is really just all about dividing your area of interest into smaller, manageable parts, called subregions or microhabitats, and then making sure that each sample of the area is represented proportionally. So you're basically combining zonation here with some zonation with quadrants, by recognizing the zones or the terrain that exists within a habitat, and then selectively placing your quadrants within regions where you're equally likely to find that terrain. So you can see here that we have less of our forest terrain, and we have more prairie. But because of that, we can, even though we have more prairie, we can selectively choose to implement more quadrants in the forest proportionally to account for that difference. We can do this using a couple of different methods. However, we don't just have to rely on quadrants. We can use line transects. This technique is super straightforward. You just lay down a line through your area and count along it. In the context of stratified sampling, you're just using this method to assess abundance within a subregion. Line transects are also great for detecting environmental gradients, like how things change as you move from water or rise in elevation, compared to quadrants where you would be working on a bunch of different scales. Line transects are convenient because they account for that differences in elevation. Next, we have quadrant sampling, like I was mentioning earlier. This is one of the best ones. It works great with stratified sampling. Once you've divided your area into subregions, you can basically just place the quadrants based on the percentage of each space each subregion occupies, like you see there on that diagram. This gives you a more focused look at abundance and makes sure you're actually capturing each part of the ecosystem in proportion to how much it actually is there. Finally, we have belt line transects, which sort of take it to the next level by combining both distribution and abundance. Basically what you do is you lay down a line and then you place quadrants at intervals around it, which gives you not only the number of organisms but also insights into how they're distributed around the area and how abiotic factors like sunlight or soil moisture might be affecting them. By using quadrants, you're able to observe differences in abiotic factors and thereby recognize how these might be related to the species that occupy those regions. We've gone through stratified sampling now, or how we can actually use it, but I'd like to quickly go over the why. So why do we actually want to use stratified sampling? Well, when it comes to estimating population, density, distribution, even tracking things like environmental gradients or zonation, stratified sampling is really useful because it considers all these factors together, unlike our previous methods. On top of that, you can calculate things like species evenness and species richness using this method, which is somewhat limited by other methods. You're not always able to calculate both using the same method. I'll go through how you might use stratified sampling to give you a better idea of this. First things first, there's actually a very set method of doing this. You need to first select your site. You do this by mapping out the boundaries of your area of interest. Once you've got that mapped, you can break it down into subzones or microhabitats. Maybe it's a patch of grassland next to a swamp or a small aquatic region. You can segregate it into small parts to make sure you're capturing the full picture of the ecosystem. Now comes the important part. You need to decide how many quadrants you have available, and because of that, choose your sampling method. So for stratified sampling, you've got to think proportionally. You need to make sure you're allocating your quadrants or placing your lines so that each microhabitat gets sampled appropriately. This brings us to a problem with stratified sampling, how you would decide what survey technique is best for the job. Your natural impulse might be to say beltline transect is the best because it incorporates both methods, but sometimes you're not looking for both. You would rather focus on a specific thing. And to make the most out of finding that specific feature, it would be better for us to use either quadrants or line transect. If your focus is on abundance, quadrants are your best bet, because you can just place them in proportion to microhabitats and then just count. You don't need to draw along a line. You're still getting a relative picture of that abundance within that ecosystem. If your goal is distribution or observing how they change relative to the environment, a line transect is really perfect for that. It shows you what's happening across your site. But there are some cases where you might want both distribution and abundance, in which case it's best to go for the beltline transect. This method basically combines the strength of both, and in turn it gives you the numbers by telling you how organisms are distributed across different zones. Okay, it's super important to keep things fair when using stratified sampling, as you might expect. You can make sure your study is big enough and has enough samples to ensure that you're minimizing bias. The bigger the sample size, the better your data will represent the whole area you're studying. That being said, obviously you can't just get an entire ecosystem. We don't live in a perfect world. It's very unlikely that you'll be able to get a picture of the entire ecosystem. So we still need to use these techniques such as stratified sampling to limit our evidence. But it's still more useful to have more data when possible. Next, when it comes to picking spots for your quadrants or transects, it's generally preferable to use random sampling over a systematic sample. So, like we mentioned earlier, you can use random number generators to decide where to place them, because then there's no chance of you subconsciously choosing good spots and messing with the results. On top of that, you can separate first into different regions and then choose random spots within those regions if you're using stratified sampling. Compare that to random sampling where you were considering the whole ecosystem as a whole. This method is making sure that you're still accounting for those smaller subregions that have different terrains. There's also the question of counting criteria. When it comes to using quadrants, however, this is a big issue because since we've made quadrants ourselves, we've put them over an ecosystem. They're not technically there in real life. We have to also decide what is considered to be within the quadrant and what's considered to be out of it. A standard rule is that if your organism is 50% within the borders of the quadrant, it counts as being within it. Again, you'll see this more as you do some practice questions, hopefully, which I would highly recommend for this topic. Finally, we're looking for a more practical standpoint here. You want to make sure all your equipment is properly calibrated and that you're paying attention to its precision. I'm sure you know why. Making sure your gear is accurate means your data will be too. If you're lucky, and depending on the school you go to, there is a chance you might get to do an excursion where you actually get to do this sampling. You could also do it at your school. So you will get a chance to ensure that, but I do want to mention that it's particularly important here because having poorly calibrated gear can completely offset your entire experiment. On top of that, you might be asked to select the method that you use in your sampling. So it's good to make all these considerations that we've made across the lesson about what the preferable technique would be to do so. Remember, it's environment dependent. Depending on what the ecosystem you're looking at is, the method will change. If you're looking at your school field, obviously you wouldn't expect to see too much variation. So perhaps a line transect would be preferable. But again, it does depend on the ecosystem. That's the main takeaway I want you to have. On that note, we're actually finished up with the content I wanted to walk through today. So really good job for sticking it out. I know it's pretty different from what you're used to in this subject. Like I mentioned at the start of the lesson, there is a lot of common sense involved. You're expected to figure things out rather than, while there is obviously a lot of memorization involved since it's biology, on top of that you are expected to use some common sense. So I know I'm really glad that you've attended today. But you've already learned content that all your peers will be unfamiliar with, so it's really setting you ahead in a subject that's very heavily reliant on memorizing things. We actually have plenty of time now to bring everything we've learnt today together and do some practice questions. So if you get through these, you'll know that you're confident going into your classes, and if not, they will be probably a great learning experience for you.